Welcome to the last lecture in the third week in the course Quantitative Methods in Chemistry. Like in the last class, we tried to understand where systematic errors could arise from. We took an example of determining the critical micelle concentration of a surfactant, and we ended up using two different techniques to see whether a given measurement is repeatable and whether it's reproducible. Of course, some numbers were given there where you were able to realize that one experiment done with conductometry did not agree with the other person. However, an independent measurement with fluorimetry was able to achieve this. What we will end up doing today is to go to the nitty-gritty details of this experiment and try to understand where systematic errors could arise from and what problems would end up arising when you do not set up your experiments carefully. And for this purpose, I'm going to be showing you the manual where stepwise clear instructions would have been written down, once again towards making sure that the experiment can be repeated by anybody across the world, and trying to emphasize in points where things could be quite different. We will also be taking a look at some video demonstrations where different steps would be performed, and we can try to also see where mistakes could arise from. So let me be also clear in the fact that we will not be describing the idea or the chemistry behind the techniques that are being used, for instance the fluorimetry and conductometry. The basic introduction that we had in the last class should suffice for us to understand whatever we are trying to do. Okay, so let's quickly recap. What we saw as an example was in the case where you have micelles that are formed from surfactants. Surfactants are chemicals that end up changing the surface properties of a liquid. In the case we are looking at sodium dodeca sulfate SDS, which has a long hydrophobic tail and a hydrophilic head which is charged. So what ends up happening is that all of the hydrophobic tail end up forming structure called micelles, where the hydrophilic outside is exposed to water. And of course there are different types of such aggregates that could be formed by these surfactants. One example is shown as liposome here, where it also has an hydrophilic interior that is formed. The micelle is one where only the hydrophobic interior is formed, where the hydrophilic exterior is facing water. And this is a very common scenario where lipid bilayer is formed in all of our cells in form of a plasma membrane, where the hydrophobic inside once again point with respect to each other, and the hydrophilic outside end up being one part outside the cell and the other being inside the cell, and entities like membrane proteins which are present in between them help transporting the chemicals. So the aim of this experiment that we saw last week was to determine the critical micelle concentration of sodium dodeca sulfate using two different techniques, namely fluorimetry and conductometry. So now, having said that, let's take a look at what are all required. As I just mentioned, we are talking about determination of critical micelle concentration for a given detergent, in this case sodium dodeca sulfate, employing two different techniques such as fluorimetry and conductometry. And as always, whenever you are setting up an experiment, it's a good idea to have every small thing that you might end up using for the experiment clearly written out. In this case, you would need volumetric flasks. You would be using 1 unit of 10 ml volumetric flasks. Beakers, you would be having different volumes of beakers and different units of them. And micropipette. Micropipette is one. Instead of the regular pipette we end up using with a rubber bulb, this one will be able to aliquot very small amounts of volume, such as something like 2 to 20 microliters quite accurately and precisely, and also something between 100 microliters to 1000 microliters accurately and precisely. As we just spoke about it, we also need a conductometer in order to perform the conductometric experiments, and other subtle things such as dropper, glass rods and thermometer in order to do its purposes of stirring and also measuring temperature in the case of thermometer. The chemical in this case that we are using is sodium dodeca sulfate. This would of course be provided in the experiment so that you can determine the necessary stuff. We will quickly understand why you would need potassium chloride in this experiment, as this would be used towards calibration of the conductometer. And the fluorescent dye, since this is a surfactant that is not inherently fluorescent, in order to perform the experiment you would have to add this fluorescent dye. And of course water in order to make up the solutions as pure as possible, in order to make sure any contaminants in water does not contribute towards error in your measurement. Think about taking tap water. If it's too hard, there could be other salts that could change your conductometric measurements and would result in systematic error there. To start with, we can try to look at how the fluorimetric experiments could be set up. The first step almost in every equilibrium experiment is to measure the room temperature at which you are working. In the case that you are working in experiments where the temperature is set at a different value apart from the room temperature, for instance, if you are performing your experiments at 37 degrees Celsius, you got to be careful such that throughout the experiment the temperature is maintained. Of course, the same problem would come up in room temperature, where as a function of the time in a given day, the temperature could vary. It is always a wise thing to you have a thermometer and keep checking what is the value or what is the temperature at which you are performing the experiment. Let us say there is way too much fluctuation. It is better off that you don't perform such an experiment at that given point of time and ensure that in a given part of the day where the temperature is set constant, you can perform the experiment. Most often labs end up having an equilibrated atmosphere in terms of an air conditioner, which will ensure the temperature doesn't change too much while you are performing the experiment. Okay, now comes the first step. As I mentioned, you will have to add 10 ml of 0.5 molar SDS solution in a volumetric flask provided to you. So this is the first step where you end up taking a stock solution. So this is the first assumption you are making. You are taking a stock solution of a known concentration. And how would have people prepared it? They would know the molecular weight of sodium dodeca sulfate. They would have measured it in a weighing balance and added a certain amount of mass to a given volume in order to make the 0.5 molar solution. As we saw in the first week of classes, how to prepare things of this sort. So first step in this experiment is to prepare sodium dodeca sulfate solution so that you can perform your measurements in fluorimetry and conductometry. The first step here would be to prepare 0.5 molar sodium dodeca sulfate. How would one end up doing it? You would end up measuring a certain amount of weight depending upon the molecular weight of the given material. In this case, it's sodium dodeca sulfate, and then you would be making it up with a 10 ml of solution in a volumetric flasks. First step that we are able to realize already here is that the mass that you end up weighing could be a reason for error, where if you are not calibrating the weighing balance, that could end up resulting in a systematic error. And on top of it, as I had introduced to you last class, every volumetric flask is referenced to a given temperature, let's say 20 degrees Celsius, and a standard deviation that is associated with it. So one has to be careful. So the error here could come from the weighing of the SDS and the making up of solution in the volumetric flask. One should remember, since SDS is a surfactant similar to your soap, if you are adding soap to water, it's going to result in the formation of bubbles, and this could result in the fact that it's not easy for you to determine the meniscus of this solution in the volumetric flask. Therefore, generally what people end up doing, instead of adding all the 10 ml to make up the solution in one shot, they end up adding just 5 ml of the solution. They shake vigorously, and in case the surfactant is incompletely dissolved, we end up sonicating it for a few minutes. So what this ends up doing is that instead of putting all the 10 ml and seeing that it's not completely dissolved, people generally dissolve salts or anything that's solid in multiple phases. So we end up adding a certain amount of salt, add a certain amount of water, not the entire amount, shake it or do whatever step. In this case, sonication is suggested. Sonication is a technique where you apply sound waves in order to provide energy and dissolve the material. So you try to dissolve it within a few minutes, and after all of the SDS has been dissolved in this 5 ml of water, then you ensure that all the bubbles that might have been created are completely taken out of the solution by using a hair dryer. So basically you warm it up such that you result in the bubbles bursting and no bubbles are present, so that you can carefully make up the solution to 10 ml. Once again, here comes the problem where if this is not carefully followed, you might introduce systematic error into your measurement. After all of this has been done, you make up the solution to the entire mark in order to get the 10 ml of 0.5 molar solution. So what are the reasons you might get systematic error here? Wrong measurement of the weight of sodium dodeca sulfate, the volumetric making up of the solution to 10 ml where bubbles could be there, or your SDS is not completely dissolved. Now, after having done that, one should be making a fluorimetric measurement, and it's quite common for many spectrophotometric experiments where the instrument is turned on for at least a certain amount of time before we start the measurement. In this case, it is recommended that you start the fluorimeter at least 15 minutes prior to your experiment. This is largely because many times the source that ends up exciting your sample has to be in its own equilibrium. Generally, these are lamps which has to warm up to a certain temperature, and their excitation bandwidth is uniform such that any measurement that you end up doing subsequently are also carefully represented in your data. So this will ensure that the source is equilibrated. Okay, now that is done. So you have prepared the solution of SDS and you have equilibrated your fluorimeter. Then the first step in this would be to dilute the fluorescent dye that has been provided to you. As I had mentioned already, SDS is not fluorescent, so therefore you have to add a dye in order to follow the measurement. This is something very similar to adding an indicator in your titration measurements. For instance, if you are doing acid-base titrations, you would end up adding phenolphthalein. And you know for a fact that unless the pH goes above a certain extent, phenolphthalein is colorless. So the basic assumption in this kind of experiments is that the indicator does not change the endpoint of the measurement. We are making a similar assumption in this experiment that the fluorescent dye that you end up using does not change the value at which the critical micelle formation happens for SDS. So this is one assumption that goes into it. While you are setting up an experiment, such assumptions are better laid out so that if there are any discrepancies, you can go back and revisit what is going on. So dilute the given fluorescent dye in a given volumetric flask, and then using a pipette, the micropipette, carefully transfer 2 ml of this solution to a clean cuvette. So why don't we take a look at how this is going to be performed. Okay, what you are able to realize here is that this is the volumetric flask we are talking about. As you are able to realize, there are some bubbles. We are assuming that the researcher has taken care of all those things, and what you see here is the fluorescent dye that we will end up using. This is the cuvette that we will be using for the measurement, and of course you always have a spare cuvette such that you can blank your solutions as required. And this is the pipette with which we will be making our measurements. As you are able to see, the cuvette is transparent on all the sides, as this is a fluorimetric measurement. If you are doing a spectrophotometric measurement using UV visible spectrophotometer, you would have cuvettes that could actually be only transparent on one parallel set of planes, not the perpendicular ones. So the pipette would help you measure very small volumes, as we had mentioned already. You have 2 different pipettes, one that will help you measure 2 ml, one that will help you measure 2 to 20 microliters. Now you are able to realize that the researcher is carefully taking out the desired amount of the ANS dye, the fluorescent dye, using the micropipette, and carefully adding 2 ml of it into the cuvette as you see here. Since it's a 1 ml pipette, we have to do the same pipetting out 2 times so as to ensure that you add 2 ml. And what could this end up happening? Let's say you have a 1 microliter error in each of the 1 ml pipetting out. This is going to end up as a 1.44 microliter due to the error propagation that we have learned so much in the last few classes. So now that is being done. One has to ensure that the sides of the cuvette are clean enough before setting up the fluorimetry experiment. And here this is the fluorimetry that we will be measuring our data from. One carefully opens the chamber such that the sample can be carefully input within this fluorimeter. We will also be seeing a top-down view of this so that you understand how the cuvette is carefully placed. As you are able to realize, the cuvette has been placed within the sample chamber. As always, with modern equipment, you would end up recording the data using a computer. Here we are just trying to show the software which we end up using in order to make a measurement. So you start the manual initialization process such that all the parameters can be carefully set. So once the initialization comes up, then you are all ready to start your measurement. So in this case, we are just going to be measuring spectra, and we are going to be measuring emission spectra. So you carefully choose what you want to choose. One must be careful in doing this, because if you choose excitation spectra, maybe the experiment is not good enough to capture the changes that you would like to catch up. And when you are doing excitation spectra, you have to be careful about the wavelength that we are using and the range at which you will be recording the data. You will soon realize that these changes are made based on the manual that we will be seeing in a moment. So the first step is to change the excitation wavelength to the desired number, which is 360 nanometers in this experiment, and change the emission window that we will be measuring between 425 and 700 nanometers. Then you also ensure that the detectors are carefully chosen. In this case, we are using 2 detectors such that it will help us get the values carefully. I am not going into the details of it, but this measurement should also be carefully done such that from these 2 detectors you take the ratio of the signal that ends up coming from these 2 different detectors. Once that is done, we end up setting it up that way. That is not important for this, but it suffices for us to understand currently that that whenever you are setting up any experiments, each parameter that you set up has to be carefully thought out and has to be put in, making sure there are no errors that propagate due to them. So once you have done that, you end up trying to measure a spectrum. So when you are able to measure a spectrum, you are able to realize as a function of the emission wavelength, the intensity keeps increasing, it reaches a certain maximum and it slowly starts to drop off as a function of the wavelength. Okay, if you might remember the last week's classes, what we ended up understanding there is that until the micelle is formed, you are not going to have the fluorescent dye that is trapped within this micelle, which will result in low fluorescence intensity. Basically the intensity maximum that you ended up seeing just a while back is going to be almost constant, but as the micelle is formed, this fluorescent dye gets entrapped within this and the fluorescent intensity will increase. So this experiment is nothing but having the ANS dye in water, measuring fluorescence and adding small portions of the sodium Dodeca sulfate, the stock solution that you have prepared, into this cuvette and see how the fluorescent intensity changes. So that is all the experiment is going to be about. And many times you will have to save your data as and when you measure your data, and this is just trying to show you as an example in a given lab experiment how you save your data. Okay, now that has been done, the next step that you would end up doing is to add a small amount of the sodium Dodeca sulfate into the cuvette. So why don't we take a look at the protocol before coming back to this. So if you are able to realize, the 2.5 is what we have done, such that you set the excitation wavelength to 360 nanometers and get the emission spectrum between 425 and 700 nanometers, and you would want to make a note of the emission maxima, which is saved in the data file that you ended up saving. Now to this 2 ml cuvette, carefully add 4 microliters of the SDS solution that you prepared in the initial step. So basically in step 2.2 you ended up preparing 0.5 molar stock solution of SDS. So carefully add only 4 microliters of it, and this is important because once again you have to do everything quantitatively. Adding 4.1 or 3.9 or 3 or 5 microliters would end up resulting in a systematic error. So one has to be careful there. Once that is done, you in theory should let the solution equilibrate for a few minutes before you do your measurements. As you are able to realize, although we are doing a thermodynamic experiment, the kinetics of formation of the micelles need not be always fast. In this case it is indeed fast, but if in case it is slow, you have to let it equilibrate for a finite amount of time. Generally something between 10 to 15 minutes for a fast aggregating system is good enough such that when we make a measurement, it's indeed in its equilibrium state. So once that is done, once again you repeat the fluorimetric measurement, you assume that the overall volume is 2 ml. If you realize you are adding 4 microliters to 2000 microliters, so it's an assumption that goes into place. So any volume that comes up, you should be carefully getting the molarity of the final concentration of SDS by using the formula that you have seen before, that is C1M1 M1V1 is equal to M2V2. So this experiment is nothing but repeating that for several different concentrations of SDS and getting the emission maximum, and then plotting the emission maximum as a function of concentration of SDS. This is exactly what we saw in the last class. Then what ends up happening is that we start getting we start finally getting data that looks of this sort. So you are going to measure concentration, you are going to get fluorescence intensity as a function of concentration. So what ends up happening, you get a constant intensity and then it increases, where at the point of break is where you are able to realize that is the critical micelle concentration, which we saw in the previous class. Now this is done, what you are able to realize, one could have errors that could come up in very many different steps. Let's reiterate that for the fluorescence experiment. It could come from the weighing of SDS, it could come from volumetric making up of the solution, it could come from the fact that the amount of ANS that you have added could interfere with the micelle concentration at which they form, the surfactant concentration at which you form micelles. It could come from the fact that when you are adding a certain amount of SDS to the solution, you have not equilibrated it enough such that the fluorescent intensity does not reach the equilibrium state that you might want to get. And it could also be the case that you might not have set up your excitation wavelength and emission spectra, the wavelength between which you want to record your spectra. So all this has to be carefully done. One other variable that we have set there as a slit width, one has to be careful how the points are digitized so that the maxima is carefully determined. So what you are able to realize that all this could result in a systematic error, and let's assume that you have done an experiment such that everything is calibrated including your pipettes and your weighing balance. You are trying to minimize the systematic errors that could come up, where only finally the random errors that are associated with the measurement itself would result in small standard deviations that would end up coming. So now that we have seen how the fluorimetric experiment works, we also would like to see how the conductometric experiment would be performed. So for this, similar protocols would exist. The first step in this would be to measure the temperature. You have to ensure that the experiment that is done in the fluorimetric condition and the conductometric conditions are in fact very similar to one another. If this is not the case, you are going to have different measurements. Remember, all these aggregates that end up forming could be subtly different as a function of temperature. We are once again imagining an equilibrium constant between the micelle and the monomer, and this could easily shift based on the enthalpy of formation of these species. So the first step that we are going to end up doing in this case is calibrating the conductometer using some standard solutions. If you remember, when we were talking about the materials required, we said we will be using a small amount of standardized solutions of KCl in order to calibrate the instrument. So let's take a look at how does this work. Just to show a little bit on how the data that we ended up saying for the fluorimetric experiment looks, when you save the data, you start ending up getting something, a wavelength and the intensity. So one should be able to plot this using different spreadsheet software or even any other program that you might end up using, such that you can reliably get the measurement that you are talking about. And if you are able to realize, since fluorimetric experiments are so sensitive, you are able to see so many decimal points that ends up coming. Just because you get so many decimal points doesn't mean it's as precise. One has to repeat the measurement a few times in order to determine what is the precision of the measurement. Now to the conductometric experiments. So this is a conductometer. This is what will help you get the reading for the solution that you have ended up preparing, that basically on what is the conductance. And if you see here, this is the probe which helps being in contact with the solution from which you want to measure the conductance. It's connected through a cable which helps this conductometer interact with the probe, which hence ends up measuring the conductance of the solution that you would like to measure. As we just mentioned, please ensure that you measure the temperature at which you are making a measurement, and it's a good idea to always put the thermometer within a water bath such that equilibrium measurements can always be taken care. What you are able to see the scientist doing here is that you are not able to read a conductance value here. This is how many meters end up working. They have their own ways of measuring things at different precision. If you are having very low conductance, one might have to change carefully the settings that go here. The first thing that one has to end up doing is to tell the conductometer that you are going to be measuring it at a certain temperature. And if you are able to realize, the scientist is actually keeping it in the temperature column and reads the temperature that is 25 degrees Celsius, and whatever temperature has been measured using the thermometer is carefully adjusted here. Since we measured 27 degrees Celsius, the researcher first changes the temperature at which you are making such a measurement. Once that is done, what he is changing it is that he is changing it to conductometric measurements, and he realizes that he reads a value that does not make sense. So what he ends up doing is to change the least count at which he can end up measuring. So what he is able to see is that when the meter is kept at 20 microsiemens measurement, you are actually not able to read anything. So what he will end up doing is to change it higher to see whether some value comes off it. So it's just changing it from 20 to 200 microsiemens, then immediately what you are able to realize is that you are able to read a value out which is 71 microsiemens. So this makes total sense, because if you are having a reading of 71 microsiemens, if you are putting it at 20 microsiemens as the maximum measurement, the conductometer is not able to read. This is a common problem that will come up with different equipment where they have their own range of measurement, and any measurements that you might end up doing higher or lower might not be detectable. So this is something that each one of us must have in mind before making a measurement. What is the correct value at which you should be measuring this? We will see more of this instance as we go forward. Of course, one can always argue, instead of keeping at a 200 microsiemens, what will happen if I keep it at 1000 microsiemens, basically 1 millisiemens. We are going to see that in a moment. So once you are able to change it to 1 millisiemen, you are able to realize that the value from 71 goes to 0.07. You are losing the last decimal that comes, which is 1 here, meaning that from two significant figures you are reducing it to 1. So you are able to realize that if you are not setting this value right, you might start slowly introducing systematic shifts that comes up due to digitization of the equipment that you end up using. So that, therefore the scientist is switching it back to 200 microsiemens in order to make his measurements. So the first thing that we would end up doing is to calibrate this conductometer. So he is adding the 0.01 molar KCl solution from the volumetric flask that he has made it up into the solution. And before you end up using the probe, the first thing that you must end up doing is to always wash the probe, irrespective of which one you are doing. Wash it carefully such that no damage occurs to the probe. Wash it enough with water and dry it with a paper that doesn't get stuck or results in scratches. So you have to be extremely gentle when you remove the water. You just touch it and it actually absorbs all the water. Then you calibrate the probe and the conductometer with the standard solution of 0.01 molar KCl. So what has to be done is that you generally have a sheet that tells what should be the conductivity of the standard solution at that given temperature, and at the back of this conductometer you would be able to adjust this value. You will soon see that the researcher will carefully change this in order to match with the tables that he has obtained. So at that concentration of KCl, the value is set to that of the literature value, which in this case is 1.47 microsiemens, rather 1.47 millisiemens. So the next measurement that he ends up doing is for higher concentration of KCl. So you are going from 0.01 molar to 0.1 molar. As you realize, conductance increases with the concentration of the ion. When you increase the concentration, you are able to realize the value went outside the detection limit at 1 millisiemen setting. So therefore what one has to do is to change the equipment such that it can carefully measure values until 20 millisiemens, and that's essentially what is being done. When you increase the concentration of KCl, conductivity increases and you are able to carefully measure it by changing the setting in the conductometer. If you have not done this, you are going to have trouble. So therefore could also result in systematic errors, and you realize that the researcher makes sure the value agrees with the literature value which we just saw a moment back, which is 13.4 millisiemen. So now that you have calibrated it at 0.01 molar KCl and 0.1 molar KCl, you are all set to make your measurements. Once again, you have to ensure that you wash the electrode and gently and carefully and dry it so that any amount of water that might be sticking onto the electrode does not result in a measurement error. Once again, systematic. In this case, let's say more water or less water gets stuck, you might have an issue where the solution of SDS that you end up adding will be more or less. Actually, in this case, it will be less because you have some water that's going to end up diluting the solution with which you are working. When you have only pure water, there is no conductance that comes. Of course, there will be a small amount of conductance which the scientist is able to change it to the 200 microsiemens value, and he is able to read about 63 microsiemens that comes up. Okay, now that is done. Of course, he is just trying to show you if the setting is kept at 20 microsiemens, you do not read anything proper. So, whenever you do not read anything proper, check whether your instrument is carefully kept at the setting that it has to be kept in. Okay, so now that is done. So this is the correct value, one has to be careful about that. Alright, now to this solution, you add a known amount of SDS that you had prepared. If you remember, that was the first step where we ended up preparing 10 ml of SDS in a volumetric flask. Now the scientist is carefully adding that solution into the beaker, and after having done that, one must carefully stir it. Although the researcher is just using swirling as the mechanism to mix it properly, it is better done with a glass rod such that this whole solution is homogeneous. And let it equilibrate again. So it is a good idea to let it equilibrate for a few minutes so that diffusion also is complete and the whole solution is homogeneous. Now that is done, you are able to realize the value of conductivity has now increased from 63 microsiemens to 79 microsiemens. Of course, he will probably try to show that if you are increasing this to 1 millisiemens measurement, you are losing the last significant figure that comes up, and it ends up rounding off the figure to 0.08, which is not a good thing. So do not do that. So one has to be careful doing this, otherwise this also results in a systematic error in the measurement. You should also not have a lower value, because that ends up creating trouble. What we have seen so far is that whichever equipment that you end up using, it's always a good idea to calibrate it before you end up using it, and you're able to realize that the rest of the protocol says the same thing. You add 15 ml of a solution and carefully add a known aliquot of SDS using a micropipette to this solution of water and measure its conductance as we have seen right now, and carefully make a note of what you end up getting. Once having done that, you're going to have a measurement that looks like this, where you're going to have a certain curve until here, and then that slope ends up changing after a point. This is of course concentration versus conductivity. So what ends up happening at this position, this value of concentration is when you start forming the micelle. So what have we learned so far? There could be systematic errors that could come in terms of the room temperature measurement. It could come because you did not make up the standard solution of KCl properly, once again coming up due to the fact that the weighing balance wasn't properly calibrated, or you had made mistakes where when you are transferring the KCl from the weigh boat into your standard flask, you may have lost some of it, so one has to be very careful there. On the other hand, let's say you are trying to add 15 ml of solution into the beaker, you add 16 ml or something slightly different from that, or when you are adding a certain amount of SDS, you add varying amounts. One time you add 39 microliters, when you add 41, sometimes 40. This ends up also resulting in the curves that don't look perfect. If you remember, the first researcher that we ended up seeing, 6.3 millimolar was the SDS concentration for CMC. We realized that that is wrong, and it comes up close to something like 7.3 or 7.4 millimolar, and that could be one of the reasons why the mistakes came up. And of course, you have to do enough measurements such that the curve looks proper, such that you can carefully estimate the CMC of that given surfactant. So to conclude this lecture, I would like to make you understand that we have seen two independent measurements of the same experiment, and one is able to realize there are subtle points that one might end up missing, which could result in overall error that is not good enough for your experiment, such that you can compare values. So one must always take care and also write the protocol in its excruciating comprehensive detail so that such mistakes do not happen. I hope this helps you understand how experiments must be set up and how to minimize any systematic errors that could come up. And the assignment that we will take a look at for this week would be setting up an experiment, and I would ask you to take a look at what all errors could prop up when you are making such measurements. Thank you very much.